Okay, so hi to everyone. This will be a presentation on the nutritional consideration for microbiota in diabetes. First, this is my disclosure, and I have no conflict of interest for my presentation today. So let me just give you the outline. First, I would like to give you an introduction to microbiome. Basically, microbiome is the presence of beneficial bacteria in our human body. Here you would understand that among all the microbiome present in our body, the gut microbiome is the most important and most well studied. Uh, so the next topic will be the gut microbiota, its role in health and in disease. Okay, the next is in understanding the role of microbiome in health and disease, give us the opportunity to look more profoundly at the potential pathogenesis of many diseases, including and especially diabetes. It also opens new possibilities for early preventions and therapeutic measures. Here we'll also touch on the what kind of diet or food that are rich in beneficial gut bacteria. So let's start with defining some important words to understand to better understand this presentation, and just to be sure that we are on the same page. Okay, first is microbiota. Microbiota is defined as the microorganism that live in an established environment, while microbiome uh, is the full complement of microbes plus their genes and genomes in a particular environment. Therefore, microbiota speaks on the collection of organisms, and microbiome speaks on the microbes plus the genes. Okay, so the third one is metagenome, which is a genetic material recovered directly from the environmental samples, while ecosystem is a biological community of interacting organisms and their physical environment. There are two types. Symbiosis is ecological interrelationship that is mutually beneficial to the involved party. For example, the gut microbiome microbes coexist in the lumen that are both beneficial to human and the bacteria themselves. While this biosis, when there is an alteration in the microbial community and its metabolites that changes the bacterial diversity to a more less diverse, so this biosis is linked to various inflammatory diseases. So the microbe cells and genome outnumber humans. Okay, the microbes outnumber cells is about 10 is to 1, meaning the human body harbors more than 100 trillions organisms compared to human cells of about 10 trillions. The microbes are mostly bacteria, but also include archaea, which is a prokaryotes, uh, viruses and parasites, and as well as fungus or fungi, which together make up the microbiota. Microbial genes also outnumber human genes by 100 times. No? If we have 2 to 20 million microbial genes, the human genes only contain about 20,000. So in terms of microbiome composition, it differs in different areas of the bodies. Here, it is represented by a pie cohort colored chart, and you can see beneficial bacteria or microbiome presence on our hair, skin, on several cavities like nostrils, vaginal cavities, oral cavities, and the gut, which is the most important. And you can see that they differ in composition as far as microbiome. Okay, so we will go to the most important microbiota, which is the gut microbiota. Okay, the gut microbiota of it's entirely composed of more than 500 different species. That's 10 to the 14th, okay, meaning 100 trillion. That's how dense the population. And the number increases exponentially from the stomach down to the rectum. The low count in the stomach is due to the hostile environment because of the low pH, um, and bacterial species also changes from stomach to the rectum. And you can see as they go down from the stomach, 10 to the 2, 10 to the 3, down to 10 to the, increases to the 10 to the 9, up to 10 to the 12 in inside the rectum. Okay, there are 4 major phyla of human gut bacteria. Okay, and number 1 is the phylum Firmicutes, which is majority, that's about 60 to 80%. Next is Bacteroides, Bacteroidetes, okay, that's comprises 20 to 30%, followed by Actinobacteria by about 10%. The lowest will be less than 1% is Proteobacteria, represented here by E. coli and Enterobacteriaceae. So when we think or talk of bacteria, we always think of bad stuff because some, but not all, are associated with infections or disease such as cholera, plague, typhoid, etc. However, there are a lot of beneficial benefit that we get from the human gut microbiome or microbiota. These are the main beneficial function of human gut microbiota. The arrow represent causal relationship. So normally the gut microbiota provide numerous beneficial function for the human host. These include, but are not limited to, involvement in substrates like lipid and carbohydrates metabolism, hormone modulations, protection against pathogens, epithelial cell proliferation, and synthesis of vitamins, especially vitamin K and amino acids. Additionally, gut bacteria break down molecules or metabolize molecules that are otherwise indigestible, including plant polysaccharides and some human milk oligosaccharides. This good relationship with the host is what we term as symbiosis, which means that the symbiotic bacteria that provide several benefits to the host as they proliferate and survive in the intestine. Now, diversity is the key to the healthy gut microbiome, as it allows for redundancy with multiple microbes capable of performing similar functions. Now, what happens when this bacteria, the invisible organism, becomes distressed? This disruption or alteration may lead to what is known as dysbiosis. And dysbiosis is associated with several diseases. And how do we define dysbiosis? It is defined as an imbalance in the microbial population and is linked to a variety of diseases, including malnutrition, inflammatory bowel disease, neurological disorder, cancer, and of course, importantly, in our field is diabetes and obesity. So there are multiple factors that may induce dysbiosis, and diet alone can impact diversity, as can medicines or medications such as antibiotics and exposure to pathogens or infections or toxins, and even persistent alteration in the circadian rhythm and many other things may result to dysbiosis. But where do our gut microbes come from? Our first dose of microbes comes from our mother. The moment we enter the world, microbes colonize our body, and depending on how and where we're born, we are colonized by different types of microbes. Now, babies delivered vaginally are covered with a film of microbes as they pass through the birth canal. So they are exposed to different bacteria. They are exposed to the vaginal bacteria like lactobacillus, provetella, and Schnitzia species. The advantage of these mixed bacteria help babies digest their meal. Okay, while babies delivered by cesarean section are colonized by a different groups of bacteria, mainly from the skin microbes, a very different sets of species. Now, the second dose will be from the breast milk. No? Okay, breast milk infant have higher population of genus Bifidobacterium and healthy strains of Rominococcus. Okay, with the lower count of E. coli, Clostridium difficile, Bacteroides fragilis, and Lactobacillus species, um, compared to a formula-fed infant. Okay. Moreover, on your right, the breast milk contain galacto-oligosaccharides, uh, known prebiotics that increases the proliferation of useful or beneficial bacteria such as Bifidobacterium, the so-called bivitogenic effect. So this is the changes in human microbiota throughout the life. No? First, the infant uh, gut will depend on the mode of birth, as we mentioned earlier. In young children, the composition of gut microbiota often varies, is very diverse, and less stable. With age, gut microbiota becomes more stable, that is influenced by diet, lifestyle, and genetics. However, in old age, the gut microbiota alters or changes and may be due to diet, lifestyle, medication, and genetics. It is less diverse compared to the younger age and present reduction in short-chain fatty acid producing bacteria, especially short-chain fatty acid butyrate, and increase in gram-negative lipopolysaccharide, or the well-known LPS that would produce endotoxemia secreting bacteria. And I will come back to that important topics later. Okay, so there's a poll question here. The question is, which of the following affects the composition of gut microbiome? Okay. Option A is the country you live in. Option B is cesarean section delivery. C, breastfeeding. D, A and C. E, all of the above. So you choose an answer. Okay. So factors that affects adult gut microbiota are the following: long-term changes resulting in lifestyle changes, geographical location and origin, antibiotics treatment, stress, and diet can modify the composition of adult microbiota, including what we have spoken about earlier, infant feeding uh, uh, and birth process, okay, in the early days of the infant. Okay, decreasing and increasing bacterial diversity, and among these factors, diet is by far is the most important factor that influence the composition and diversity of the intestinal microbiota. So again, I'm showing you the beneficial function of gut microbiome. Emphasis on now on the mechanism on how the gut microbiome protect against pathogens and toxins. So as a backgrounder, let's talk about intestinal barrier, meaning from the gut lumen to systemic circulation. Epithelial cells or enterocytes constitute a physical barrier, okay, impeding the translocation of luminal content of the inner tissues. As illustrated in these slides, the barrier is not only physical but also microbial layer. Okay, the gut microbiota. Okay. Chemical is the mucus layer. The thicker the mucus layer, the better is the barrier uh, that is above the epithelium, and below the epithelium, it's our immunologic, our sentinel cells in the innate immunity, as well as muscular barrier below this, as I've said, below the epithelium. Now, microbiota and immunity, it has a good relationship between, this is the relationship between gut microbiota and our immunity. So gut microbiota plays a fundamental role in the induction, training, educations, and functions of the host immune system, both innate and as well as adaptive immune system. So the training for defense against pathogen, that's in A, and train to be tolerant with non-pathogens or our own self cells, okay, also our own cells. And this is a delicate balance between the two. Any disruption, which may mean dysbiosis, therefore, leads to autoimmune disease such as type 1 diabetes and metabolic disorders such as metabolic syndrome, obesity, and type 2 DM. Now, let's now go to pathogenesis of type 1 diabetes. And we all know that it is both genetics and environment. And there are environmental factors or triggers that leads to immunologic response targeting the beta cells. And here you can see that the both genetics and environment component and the putative environmental factor on your right, which include viruses and certain proteins and toxins. So let's talk on the hygiene hypothesis. And what is hygiene hypothesis? Uh, Hygiene hypothesis suggests that a lack of early childhood exposure to infectious agents, symbiotic microorganisms such as gut flora, uh, viruses and parasites increases susceptibility to allergic and immune disease by suppressing the natural development of the immune system. In particular, the lack of exposure is thought to lead to defects in the establishment of immune tolerance. So meaning, uh, hygiene or too much hygiene in early life may cause defect in immune tolerance that leads to autoimmune disease, including type 1 diabetes. But I'm not sure if this is feasible or workable during pandemic. Nevertheless, this was the hypothesis. But how does the gut microbiota relates to the immunity and development of type 1 diabetes as an autoimmune disease? The immune system processes antigen that pass through the gut, and this is a pivotal to immune tolerance, immune recognition, whether itself or not, and in some cases, a profound immune response to those antigens that may lead to autoimmune like type 1 diabetes. Okay. So here's another poll question. Okay. What are the leverage points to enhance the role of microbiome, thus affecting the development of type 1 DM? A. Sterilizing the gut with antibiotics, kill all the microbiome. Okay. B. Giving beneficial microorganism orally. C. Giving vitamin D. D. B and C. And E. All of the above. Okay. Choose your answer and we will go forward. Okay. Okay. This is tight junction. Okay. There are several known intestinal proteins that serve as a gatekeeper or barrier function of the tight junction, including occludin, claudin, jam, etc. Okay, some of these protein components are targeted by viruses, okay, such as adenovirus, Coxsackie virus, etc. And if destroyed, may lead to leaky gut. So what is the role of vitamin D? In the intestinal epithelial cells, the active form of vitamin D serve as the primary ligand of the vitamin D receptor, the BDR, abbreviated here. The activated receptors then activate or binds to the genes, the vitamin D responsive element, and activated receptors, vitamin D receptor also regulates innate immune response, uh, activating or synthesizes defensin B, which is an antimicrobial activity, especially as an antiviral. The active vitamin D also play a role in maintaining intestinal barrier function, preventing leaky gut. Moreover, vitamin D stimulate and upregulate uh, T helper cell 2 and T regulatory cells that leads to several anti-inflammatory interleukin or cytokines and downregulate pro-inflammatory cytokines, meaning decrease pro-inflammatory cytokines and increase anti-inflammatory cytokines by vitamin D. Uh, this is a comparison between healthy GI epithelium and a thick mucus layer, and you can see produced by the microbes. Okay, and compare this to an exposure to diet that are rich in gluten, bovine milk that leads to dysbiosis, and this produces less mucus layer, disruption of tight junction, all leading to leaky gut, causing inflammation, decreased insulin sensitivity, etc. So these are the factors associated with type 1 DM. Reduced risks are the following: breastfeeding, vaginal delivery, dirty environment, bacterial and parasitic infection. Okay, while increased risks are viral infection, C-section, hygiene hypothesis that we talk about, which state that less exposure to parasite and microbes during early life leads to an overactive immune system that leads to autoimmune disease like uh, type 1. Stress, low vitamin D, cow's milk, and gluten. So in theory, probiotic, by increasing uh, bacteria, this is a probiotic contain cultures of so-called good bacteria, uh, improves or reduce permeability and maintain tight junction of gut epithelial cells. In addition, these bacteria ferment plants oligosaccharides to produce small chain fatty acid, uh, especially increase in butyrate, and maintain the tight junction and increase mucin uh, synthesis. So, is there any study looking at probiotic effect on the development of type 1? And there is one, it is the TEDdy study, or the environmental determinants of diabetes in young. The objective is to examine the association between early probiotic exposure and islet autoimmunity among children in TEDdy, which are uh, children that are genetically predisposed to type 1, has a genetic predisposition to type 1 diabetes. So early probiotic supplementation at age 0 to 27 days was associated with a lower risk of islet autoimmunity when compared with probiotic supplementation after 27 days or no probiotic supplementation. So the conclusion in this study says that the early probiotic supplementation may reduce the risk of islet autoimmunity in children at a higher genetic risk of type 1 DM. But this is a small study, but we need a larger study to prove the real benefit of probiotic in this class of patient. Now, how about breastfeeding study? Okay, there are two large Scandinavian birth cohort study coming from Denmark as well as Norway, and from over 155,000 mother and child pairs. The lack of breastfeeding was associated with a 229% increase in the likelihood of developing type 1 diabetes. So there you are, the association of breastfeeding with a lower chance of developing type 1 diabetes. Now look at let's look now on type 2 diabetes. Now modern lifestyle wreak havoc on the microbiome. All of this contributes to the dysbiosis. Cesarean section, stress, excessive hygiene, physical activity, or physical inactivity, uh, high processed food, high fat, and low fiber diet, more importantly, that may uh, give rise to obesity, insulin resistant, and type 2 DM. Now, let's look now on the microbiome and the onset of metabolic syndrome as well as obesity. Okay. We all know that type 2 DM usually start with obesity and metabolic syndrome. Okay. So comparison of lean versus obese individual as to the gut microbiome, gut epithelium, and metabolic outcome. Dysbiosis lead to increase in lipopolysaccharide uh, presence in the uh, gram-negative bacteria cell wall that causes reduced tight junctions, leading to leaky gut, increase in systemic LPS or lipopolysaccharides, lead to what is known as endotoxemia and produce chronic low-grade inflammation that we see in many metabolic diseases, including insulin-resistant obesity and type 2 diabetes. Moreover, dysbiosis causes a decrease in energy harvest for the epithelium. Okay, a metabolic outcome can increase in insulin resistance, LDL, increase in lipids like LDL cholesterol, and triglycerides, and obesity, or adiposity rather. So again, what is the leaky gut? This is when the gut barrier deteriorates and the toxins from the various bacteria as well as other products may leak into the circulation or systemic circulation from the gut. Okay, so what is the mechanism? The mechanism of leaky gut is the encroachment, no, of microbiota on the enterocyte. The distance, and you can see the distance from the epithelial or enterocytes to the bacterium becomes narrower with a thinner mucus layer compared to the controlled patients. No? And this microbiota encroachment is the future of metabolic diseases, particularly insulin resistant, associated with dysglycemia. Okay, but how does it cause insulin resistance? Again, these bacterial product changes in adipose tissue lead to insulin resistance and decrease in insulin release. How? By number one is increased fat and sugar by Western diet, increased bacterial release of this LPS, and the LPS now produces or leads to the inflammatory cytokines into the portal system, um, as well as to the systemic system eventually. And number three, increased translocation of bacteria and LPS into the visceral adipose tissue, causing increased inflammatory cytokines. Number four, the adipocytes releases a lot of free fatty acids. Number five, reduced clearance of inflammatory mediators from the visceral adipose tissues. And number six, increased LPS, free fatty acids, and the cytokines into the circulations, decreases liver metabolism and insulin sensitivity. And lastly, number seven, increased delivery of LPS, free fatty acids, cytokines into the systemic circulation negatively affect our beta cells and systemic insulin sensitivity. So under healthy condition, as you can see, the lean phenotype against the obese phenotype, you can compare as far as the alteration of gut microbiota. In lean, there is symbiosis. In obese phenotype, this is dysbiosis. In the lean, there are controlled pathogens and increase in butyrate producers, okay, of bacteria. While in the obese phenotype, expansion of pathogen, loss of butyrate producer, okay. As far as uh, small chain fatty acids, there is a balance with the lean phenotype, balanced energy harvest, appropriate immune response, anti-inflammatory microbiota, and tight intestinal barrier. In contrast, in the obese phenotype, there is disturbed small chain fatty acid profile, increased energy harvest, disturbed immune response, pro-inflammatory, and leaky gut. So altered microbiota, or what is known as dysbiosis, is generally characterized by expansion of usually underrepresented microorganisms, often opportunistic pathogens, and lower phylogenetic diversity. Okay, so this is the intestinal barrier that is which is disturbed in people with obesity and diabetes. Okay, a high fiber diet supports intestinal barrier function by improving intestinal tight junction expression in the normal immune cell and expression of the normal immune cell function, as you can see in the lean on your left. Okay, the intestinal integrity is affected uh, in people with metabolic syndrome. That's seen on your right. They have a thinner mucus layer, as you can see, and that leads to penetration of opportunistic bacteria, lower level of IgA positive B cells, and lower IgA secretion, which may end into microbial alteration or overgrowth or opportunistic pathogen. This is a Western diet that increases intestinal tight junction expression, which result in translocation of bacteria and pathogens associated molecules. Okay, high glucose levels reduces tight junction expression via GLUT2, promoting bacterial translocation in people with diabetes. Okay, in particular, adipose tissue macrophages, okay, um, are responsible for low-grade inflammation, high, meaning high pro-inflammatory cytokines levels, and less anti-inflammatory cytokines. So, what's the benefit of short-chain fatty acid? So, high fiber diet leads to fermentation of short-chain fatty acid uh, by the microbiome, a gut microbiome. These short-chain fatty acids binds to to various G protein coupled receptors that are expressed in the enterocytes, as you can see. This leads to secretions of hormones such as PYY and GLP-1, and these two hormones improves or increase satiety and improve insulin sensitivity. Moreover, uh, the short-chain fatty acids can activate G protein coupled specific G protein coupled receptor, which is GPCR43, that is expressed in the adipose tissue. When it is activated, it reduces insulin signaling and reduce fat accumulations. Now, the microbiota with an individual with high fiber diet also sufficiently suppress the fasting-induced adipose factor, or the FIAF. Okay. Okay. So this expression in the lumen, which inhibit lipoprotein lipase activity and fat storage in white adipose tissue. So these are all the benefit that you the short chain fatty acids that has a that gives you the clinical benefit when you take high fiber diet. Now, now again, we look into the endotoxemia insulin resistant with high fat diet. Okay, this time it elevates plasma lipopolysaccharide, or LPS. Now this LPS, as we mentioned earlier, reduces gut barrier functions. LPS is present in the gram-negative cell wall. If you take a lot of high-fat diet, you would have overgrowth of these bacteria, and the LPS triggers inflammation, damages beta cells, and inactivates insulin receptors. Okay, so um, we talk on the nutrition and microbiota, so eating pattern and food groups. Okay, in the Western eating pattern, it has shown that it's altered the genetic composition of and metabolic activity of the human microbiome, and this is a significant contribution to modern diseases. No? While high fiber diets, such as whole grain, fruits, vegetables, and legumes, have been found to induce a more diverse microbial population, which is beneficial. Significant differences in terms of microbial composition and health have been demonstrated between plant-based eating pattern and Western eating pattern. This was demonstrated in the study done by De Filippo et al. Comparing fecal microbiota of rural 14 African children from Burkina Faso, Western Africa, and 15 European children aged 1 to 6. The Burkina Faso diet comprises of cereals, legumes, and vegetables, while European diet comprises mainly of animal protein, high sugar, starch, and fat with a low fiber diet. So the summary shows that Burkina Faso children increases bacteroidetes, increase Prevotella, and increase that this microorganism, okay, increases the genetic diversity and increases fecal, okay, short chain fatty acid production, compared to the European children, which increases Firmicutes and Bacteroides, and these bacteria decreases the genetic diversity and fecal short chain fatty acid. Now, what is a prebiotic? Now, prebiotic is dietary fiber. It is not digested or absorbed in small intestine. Therefore, it is fermented by the gut microbiota, okay, usually in the colon. It stimulates growth of health-promoting species like lactobacilli and bifidobacteria. So these are examples of prebiotics: breast milk, artichoke, uh, chicory roots. We don't see that much here locally. Okay, leeks, onions, garlic, asparagus are very common. Whole meat, beans, banana, oat, soybeans, psyllium, husk, which is can be marketed, can be both commercially under the name metamizil. So, what are probiotic? The definition of probiotic is pro is for four and bios is life. So, live microorganism it colonize and provide health. It should provide health benefits. Okay? So, what is the criteria for probiotic classification? So, it should resist gastric, bile, and pancreatic digestion. Okay? Adhere to the mucosal and epithelial surface. Once colonized, it should remain metabolically active and provides the host with protections and pathogens by secreting antimicrobial substances or preventing their adhesions and colonization. These are food rich in probiotic. They're divided into plant-based sources and dietary-based sources. Plant-based are sauerkraut, kimchi, and etc. Uh, dairy dairy-based are yogurt and kefir. Lactobacillus is an example of a commercially available probiotic is Lactobacillus casei shirota strain. And many fermented foods, no? Such as yogurt, sauerkraut, and kefir. Uh, However, the most recent interpretation of definition has included these as traditional ferments. Okay, and fermented food are now considered food sources of live and active cultures, but not probiotics. Okay. So yogurt uh, is one of the very common food stuff that contains a lot of probiotics. So these are fermented. Okay. Kefir, uh, not very common locally. It's similar to yogurt. It's fermented milk drinks that originated from Caucasus Mountain, that's in uh, mountain system in West Asia between Black Sea and the Caspian Sea in Caucasus region. Okay, uh, okay. So countries mainly Armenia, Azerbaijan, Georgia, and part of southern Russia. Uh, they have kefir and yogurt. So fermented vegetables that I mentioned earlier are very rich in probiotic. Uh, Sauerkraut is a fermented product made from cabbage, and I think it has roots in Central Europe. The Germans and some regions in France are uh, have a lot of sauerkraut. Kimchi, Korean kimchi, and probiotic supplement. Okay, um, are not regulated by FDA, often not tested. Okay, there are a lot in the market. Generally are considered safe, but look for USP GMP certification. Take with food, okay, 2 hours before or after antibiotics because they can now antibiotic can alter the growth of or kill the bacteria if it's simultaneously taken. Changes are transient. If you stop it, meaning you have to continue taking the probiotic supplement. So, is there any trials looking at probiotic in type 2 diabetes, its effect on the there are two they look into the effect on the markers of diabetes control, inflammation, and oxidative stress. And one study is a Brazilian studies and one is Iranian study, and they showed that it's an RCT, although very limited numbers of patients. Okay, uh, they found out that it reduces fructosamine, hemoglobin A1C, even lipids, LDL cholesterol is reduced, as well as inflammatory cytokines. Um, on the Iranian study, they showed that there were reduced serum insulin, reduced fasting glucose, C-reactive protein, and also increase in reduced glutathione, which is an antioxidant. So that's the end of the presentation, and I'm open to any questions regarding the presentation. Thank you for listening.